Gases and biochemical reactions. First, let's look at what a biochemical reaction is. Biochemical reaction. Biochemical reaction is any reaction that takes place in a living organism. Reaction that takes place in a living organism. So, all living things need a constant supply of energy just to stay alive. Everything that a body does depends on these biochemical reactions. And these biochemical reactions are reactions that take place in a living organism, Respiration is one of the most important biochemical reaction in humans and in plants. Photosynthesis. Photosynthesis is a very important biochemical reaction in plants. Gases also play an important role in everyday biochemical reactions. Respiration requires an oxygen gas and produces carbon dioxide gas during the reaction. So the chemical equation for respiration is C6H12O6 plus this is the chemical reaction for respiration. C6H12O6, this is glucose from food, oxygen, 6CO2 plus 6H2O with some energy. This is the reaction for respiration. So it uses oxygen gas and produces carbon dioxide gas. This carbon dioxide is part of the carbon cycle. So a biochemical reaction is a chemical reaction that takes place in a living organism. Metabolism. Metabolism, we've already looked at metabolism before. So metabolism. This is all the chemical reactions that take place in a living organism. So it's a sum of all the chemical reactions, a sum of all these chemical reactions that take place in a living organism. So metabolism is the name given to all the biochemical reactions working together in every cell of our body. Trillions of cells undergo thousands of reactions in a single day. That's a lot more chemical reactions than scientists do in a laboratory. These biochemical reactions often need oxygen gas as a reactant. Carbon dioxide gas forms as a product. So all the things this side on the left side are called reactants of a chemical reaction, and the things on the right side are called the products of the chemical reaction. So in respiration, the reactants are glucose and oxygen. Products are carbon dioxide, water, and energy. Many reactions occur, so the rate of reaction needs to be very high. Our bodies have different ways of ensuring that this rate is high so that our bodies can function properly. So, gases are very important in our everyday lives. Without oxygen gas to breathe, we could not survive. Plants and trees could not survive without carbon dioxide gas. Hydrogen gas is often used as a fuel source. It's used to launch rockets into space and in some new vehicles, and produces no harmful gases when burned. So, these two biochemical reactions we're going to talk about are respiration and photosynthesis. the chemical reaction for photosynthesis, 6CO2 plus 6H2O in the presence of sunlight and chlorophyll gives rise to C6H12O6 plus 6O2. This is the balanced equation for photosynthesis, and it takes place only in plants. So respiration and photosynthesis are two very important biochemical reactions because they provide energy to the living things. These chemical reactions use a cycle of gases, oxygen, carbon dioxide, and water vapor. Now, let's look at catalysts and enzymes. Let's look at what a catalyst is. A catalyst is something that changes the rate of a reaction. It's something that changes the rate of a reaction. Changes, or we can say alters. Or it alters the rate of a reaction. Catalyst can increase the rate of reaction or decrease the rate of a reaction. Now, enzymes. Enzymes are biocatalysts because these are used for biochemical reactions, and these are the catalysts that are present in living organisms only. So enzymes is what we call them. The reactions in our bodies need to happen very fast. So we already know that enzymes are biocatalysts that speed up biochemical reactions. We learned that increasing temperature or concentration speeds up a reaction. The cells in our body would not survive a big jump in temperature. This is why enzymes are so useful for biochemical reactions. So now let's look at enzymes and their properties. Enzymes. First, enzymes are proteins. A very important thing to remember is that enzymes are proteins. Next, enzymes do not get used up in a reaction. They do not get used up in a reaction. And they don't make more of a product. Don't make more of a product. So they do not get used up in a reaction and they do not make more of a product. The next thing, enzymes lower the activation energy. So activation energy is the energy required to do a chemical reaction. So these enzymes lower this activation energy and it makes the reaction much more efficient. So they lower the activation energy for biochemical reactions. Also, they're very specific. Very specific. So certain type of enzymes work for only certain types of chemical reactions. So these are very specific to the reaction they are needed for. And they speed up the same reaction over and over again, and because they do not get used up in a reaction, they can be used over and over again. So, in our bodies, hydrogen peroxide is often a product of biochemical reactions, and a buildup of hydrogen peroxide can be toxic for our body. So our bodies use enzyme called catalase to break down hydrogen peroxide into oxygen and water. So H2O2, that's hydrogen peroxide, use and uses an enzyme called catalase, and it breaks down hydrogen peroxide into oxygen gas and water, O plus H2O. And this occurs in the liver. So catalase is present in liver, so that's why. That's why it occurs in liver. Now, let's talk about collection of gases. When we produce gases in the lab, we need to be able to collect them. Collecting gases can be useful if you want to identify the gas being produced, or if you want to stop a gas from escaping the room, calculate the rate of reaction, or compare variables relating to the production of the gases. So there's 4 ways we can collect gases. 4 different ways. Collection of gases. The first way is called upward displacement. And the second one's called downward displacement. The next one is called over water. And the next one is using a syringe. So, we can use upward displacement and downward displacement if we know about the gas being collected. So for example, if a gas is denser than air, then we can use the method called upward displacement. So the gas being collected during this method is denser than air, and so will displace air up and out of the jar. Gas should be denser than air. Example of a gas which is denser than air is carbon dioxide, CO2. This is denser than air, so that's upward displacement. Downward displacement, here the gas should be less denser than air. Should be less denser than air. Example, helium. So helium is found in balloons. So that's why balloons are able to fly upwards, because they're really dense, they're really less dense than air. So the gas being collected is less dense than air and will rise to the top, pushing the air down and out as a result. Over water is, here the gas will bubble through the water, pushing the water down and out of the test tube. And using a syringe, the gas will rise from the conical flask and into the syringe by pushing the plunger backwards. So we can draw a little diagram of using a syringe. This method. This is a conical flask. This is the tube. And this is the syringe. And this is a plunger. So we can label it. This is the syringe. This is the plunger. This is the conical flask. And here is where the reaction occurs, in the conical flask. So reaction, producing gas. And this gas collects here. So what happens is the gas will rise from the conical flask and into the syringe by pushing the plunger backwards. So the gas gets collected in the syringe here. So the next one is over water. So we can draw a little diagram for this as well. Again, we have the same setup, a conical flask. And then we have a small container with water. And then we have a test tube. This is a test tube. And this gas collects in this test tube. So gas collects here. And so this is the test tube. And this tube is connected to this test tube. And this is the same thing as this. So here the reaction occurs, which produces gas. So reaction producing gas. So here what happens is the gas will bubble through the water, pushing the water down and out of the test tube. But the most important ones are upward displacement and downward displacement. So these are a few ways of collecting gas. So gases that are less dense than air rise, gases that are denser than air fall. Based on the densities, we can choose the best method of collecting gases produced. This can be difficult to determine if we do not know the density of a gas at the beginning. Though, if we do not know the density of a gas, we can collect it over water. If the gas is soluble in water, we can use a syringe to collect it. Collecting gas with a syringe can be used for any gas and also gives a more accurate reading of the volume of gas being produced. Now, let's look at the test for gases, how to distinguish between gases. So, distinguishing between gases. So we know that gases are tasteless. So we are only talking about three important gases here. So hydrogen, oxygen, and carbon dioxide. So we know that these three gases are tasteless. Odorless, no smell, and colorless. So how to distinguish between these gases? We have different tests for these gases. For oxygen, you have to put a lid splint into a test tube. So if you want to know like if there's hydrogen present in a test tube, you have to put a lid splint into a test tube, and then a pop sound will be heard. A pop sound will be heard. So if a pop sound is heard, that means hydrogen is present in that test tube. For oxygen, same thing, put a lead splint, put a glowing splint into a test tube. A glowing splint into a test tube, and then this glowing splint will relight. So if it relights, that means oxygen is present. So put a glowing splint into a test tube of the gas into the test tube, and the splint will relight if oxygen gas is present. For carbon dioxide, it's a bit different. So basically, you have to bubble some of the gas into a tube of lime water. So what happens is this lime water will go from colorless to a milky color, so like a cloudy milky kind of color. milky color. So, and this happens if carbon dioxide gas is present in that test tube. So these are 3 different tests that we can use if we want to know which gas is present and if we want to like distinguish between these gases. So this is about gases and biochemical reactions, and there is lastly another technique, another method called electrolysis. So what's electrolysis? Electrolysis is an industrial method of making hydrogen and oxygen gas from water. So it's basically separation of water into hydrogen gas, and oxygen gas. So basically what is done in electrolysis is electricity is passed through water, H2O, and this splits the water into hydrogen gas and oxygen gas. So this is what electrolysis is, just a basic definition of electrolysis. It splits water into hydrogen gas and oxygen gas, and basically electricity is passed through this water, and this splits into hydrogen and oxygen gases. So in this video we've looked at gases and biochemical reactions.